Yeah. It's fun. Yeah. And then consistency very hard. We will gather yeah. Yeah. Yeah, yeah. So it's already membership. But yeah, we can have an extra. We can have on the website. We sign, read, and gets 2 documents so that we oh yeah, 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 yeah. 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 Even though it's going through it and I was wondering, so you know, I'm always looking for it. And so I always go back to my feedback and to the character because it's really like I was thinking maybe Steve, I kind of like this. Yeah, I really good to do that. I would say it could be cool. And how it's yeah. And so again, and I wanted to have yeah, yeah, yeah. Yeah, fake them. You know, you can like yeah, you can really write a little bit. I have a good it's so we can talk about some food. Yeah. We have the extra. I just can't get into oh, yeah. Yeah, but like, they have a for very That's good.
So
I think we can leave this for a bit, right? We can do.
if you think this time, I think we can wait till it's the time to start, and then we can take a
Yeah, and then we can take a break and then we can set it. Also, I think we should start like that some of our brothers what we don't know.
This one?
Very efficient when we work together. It's very hard. I do the self help. I see blue. Oh, yeah, it's not on the break. That's
Hello, everybody. Welcome to Media Lab tonight. I'm Li Yao. I'm currently a PhD student on the third floor at Camping Group. This is today, my colleague today. And we are very honored to co-host this event together with the BioChange initiatives. We were contacted by Stefan about a month ago, and he mentioned this amazing idea of hosting a BioChange event in the Media Lab. And we got contacted because of one of the projects we did, which we will talk about later tonight. It is related to biology. But we got excited about this actual organization of the events because our passion of trying to reach out to the larger MIT community and also potentially the larger community within Boston, within the US and beyond the US to the world.
So it has been a big hot topic of engineering, design, biology in the Media Lab. Lots of groups in the lab actually like started designing the project, starting their own interpretation about how biology can intersect, for example, computer interfaces or semiconductor or architecture and so on. And actually, if any of you knows a little history about the media lab, that the founder of Igor Pantin used to say that being digital, like how digital is important to the media lab. But now recently he actually stated bio is the new digital. And so we hope that through these events, and then we can start a platform that foster a broad communication between departments, between researchers, faculty, and students, and think about how to situate biology in science, engineering, design, and art. Yeah. So let's move to.
So, BioChanges is an internationally collaborative community of academic researchers, industry professionals, and professional designers. And 2-3 years ago, we started working with myself, Dr. Dominic Southgate, Dr. Laura Alonso, and Dr. Victor Mujita on this idea of building biodesign facilities in London. And at that time, we wouldn't really have much interest in doing it. So instead, we started working on different projects. So one of the projects was using this 3D bioprinter, which I will talk about later. But we also worked with Raymond McCauley in BioCures, who's also the head of biotech in Singularity University now as well. And the idea was at some point to build up all of these communities and have international facilities across the world. That could be linked to the biomaker space as well. So then in 2015, Dominic introduced me to Professor Peter Clouds and then Professor Morris Pennington. And they brought me along to the Royal College of Art and Design to teach the innovation design engineers kind of biodesign concepts, and or as Miles kind of coined the phrase, mystifying methodology in bioengineering. They also wanted to meet with all heads of department to actually build these facilities within the Royal College of Art and Imperial College London. And these are actually going ahead with Professor Oran Katz, who is a visiting professor in the RCA and is setting up one of the labs in the Royal College of Art, and the advanced hack space in Imperial College is also setting up another facility, and hopefully they'll be linked. In addition to that, we started working with NUI Galway and Professor Charles Spillane wanted to set up a lab where they could allow designers and artists to come in and use their spaces. And around the same time was when I met Hiroshi. And when Hiroshi and Media Lab presented at the Royal College of Art, it kind of sparked an interest outside of everyone that was in innovation design engineering. And so like fashion designers and textiles all wanted to learn more. So that was when we started the first BioChanges event, which was in the bar in the Royal College of Art. From there, we moved to another bar in Imperial College. And from that bar, we went to the Alexander Fleming Hall in Imperial College and now the MIT Media Lab. So it's kind of progressed from bar to lecture hall, and we're still working on building these facilities, but also building upon the community internationally. So not just in London and Europe to actually connect over to the MIT Media Lab, because I worked with Charles Bracchia back in 2013 on this concept as well. And so from there, we're still building on it, helping them set up the facilities, and also a lot of projects that kind of started. So there are a few projects at the moment. One is actually, they're all looking for collaborators in MIT or even here. One is actually exploring the acoustic properties of biomaterials for novel applications. Then there's the biofashion project, which is using, I guess, shape memory polymers and biomaterials for kind of responsive garments. And while we were setting up the biodesign courses with innovation design engineering, we were actually running these microfluidics workshops, which are also being run here through David Palm and Head of Almost Anything. So it's kind of very similarities between what we're doing on both sides of the pond. Another project, this is at Central St. Martin's. One student is actually trying to work in leather. She's trying, building up facilities to do it, but she's done kind of the basic concept behind it. And then another one is actually merged bacteria with graphene to kind of create conductive skin. And that's similar enough to an IDE project where they've done something similar for bioelectronics. Philip from BioFeatures wanted to show this as well, because it's such an interesting technology that his device is like a bento box where you can kind of centrifuge and extract DNA and then use PCR and get electrophoresis on something that is, like the size smaller than the laptop. And they're starting to kickstart in March. And Philip actually set up a BioFeatures event space in London, which allows people to be trained on all these techniques as well. In addition to this week I was actually talking to a group in Peru about setting up these floating labs across the Amazon. So that's another project we're trying to work on with the Fab Lab as well, and also the network in Latin America. It'll be interesting to see how that comes along. These are all of the people that we have worked with so far. So people that have either spoken at the events or partnered up with us, including the MIT Media Lab at the top. And it's kind of, we're all working together on the same goal to try and expand what we've done with biodesign innovation here and in London. And I will talk about the company that I'm co-founder on, which is Robotics. So Robotics is a company that was founded to kind of change the way bioprinting is at the moment and kind of revolutionize it. Meaning behind Ouroboros is that it symbolizes the sense of kind of constantly recreating itself and Egyptian times, and then you've got one of our design advisors there, Dr. Southgate, kind of representing this is that we can create this technology and work on different materials or software, but we need to constantly reinvent it, which is what we're doing with some of our academic partners. So Gemma started working on these kind of basic bioprinters around 2011, 2012 in her kitchen and her home, and as you can see, they're pretty rough models. I was doing the same thing at the time, but we kind of both saw well they work, but they're not that great. So this is one of the projects for the Rio Tinto Sports Innovation Challenge, which was headed up by Dominic Southgate. And we used their conductive ink and the bioprinter, it's a basic bioprinter to actually to print this conductive mat. And as you can see, it's a pretty basic machine and they're quite easy to get a hold of. So this was done 3 years ago. And then since then, I've seen other companies come out with the same concept. And so before, even though Gemini have a more advanced machine, we kind of thought that we should release an open source 3D bioprinter. So we're not actually wrote the protocol for an open source 3D bioprinter, which is the basic one. It's on and Sims, helping behind the case. And the idea behind this is that we're actually planning to use this as a model that we'll open source and let anyone use, because the parts cost around 200 £300, but also if people want to buy it and maybe we run a Kickstarter, or since the head is so basic, we might actually get another novel extrusion head. But the idea is that it's to demonstrate that these bioprinters are quite, some of them are quite basic machines, and they should be available to almost everyone, the environmental communities or children or people in the education system. So at the same time as that, we had Gemma was kind of working on these early designs for multi-material and durable bioprinters, and they function essentially like the star arm. And so using these designs, she then took the company to HAX. And so from January until May, kind of started to develop the company further. And so we were able to print in liquid and then just do regular 3D printing. Then in July, we started to build the lab, which is essentially a biodesign facility. And by September, we had it up and running. And then, even though the slides were a mess, by January this year, we have this kind of novel retoolable multi-material bioprinter. The idea is that it can do 10 multi-materials. It's got a swap and material system. You can print cells, biomaterials, and shit and alloys. It prints in air and liquid. It's completely upgradable, modular, and only 13,000. So it's a short video of it working. So it just functions like a robotic arm where it comes in and it can sense anything. In this case, it's sensing the syringe. So it's printing using the syringe. But in any case, that can be any tool head there, and it could be printing like that and functioning like a robotic arm. So as of last week, Gemma won the award for the most innovative startup in Europe. I was actually in Dublin, and we got some money for AdWords from Google, and then we built this also kind of novel incubator in Google, which we're still working on, but the idea is to have that as a separate attachment for kind of physiological work. And this is the main team at the moment. That's Gemma on the left. And these are kind of another text we merged together. But this is these are kind of our advisors, so they're all quite well established in their fields. And so we've got Professor Alexander Spivey, who has actually created the birds for track ages in the picture there. And he diagnosed Brendan Corker, who works on veterinary medicine, Jim Moore, who works on microphysics and medical device design. Danny Green, who has his company, who is also a principal fellow in entrepreneurship at the Imperial College. Bruce Silva is a professor of tissue engineering. And Kevin is a co-founder of the Science Foundation, and Dr. Dominic Secchi, who is from BioChanges, is also a senior teaching fellow in design engineering. And then James, who's hanging around here as well, is an expert in regenerative biology. Bill Yao is a co-founder of InBio, and Dr. Ravi Goliath and Trevor Kerr are both lecturers as well. And so a lot of these people are our advisors, but we're also working on interesting projects that they're working on in their fields. And one of the most interesting at the moment is using kind of robotic sensors in cartridge at home. And then so the idea is that kind of future bioprinting is what it will actually be. And it's actually slightly else. But so I guess it's email me just use the BioChanges at gmail.com. Because otherwise you're not going to read that. Yeah. Thanks. And I guess we'll go through questions at the end because we've got a few presentations to actually get through. And we'll take a break before Julie's kind of keynote talk.
All right, good evening. Thank you for inviting me. I'm really pleased to be here. I have a nice chance to tell you about a project that I run called BioBuilder. And if you remember only one thing from the things that I will say over the next few minutes, it is that I hope you will find some place that you might be interested in joining us as part of the work that we do with BioBuilder, something that is of interest to you. Please know that we are really empowered and driven through, contributions of the community and the work that people might like to do within the biobuilder community that you're engaged in. So if something is of interest to you, please do reach out, let me know, and we can try to find a project and get some work done. BioBuilder is a nonprofit organization that I founded a handful of years ago with the notion that we do not usually bridge the interesting work we do at the academic university level with the kind of teaching that we do in K-12. So my hope is to really bring, to change the way our classroom lessons are taught to better reflect the kinds of exciting things that are going on in science and engineering at the institution of higher ed and industry. So what I will talk about are things about the BioBuilder mission. I'll give you an example of some BioBuilder content that we provide, and then talk about some of the future endeavors that BioBuilder is planning. So in terms of mission, what I think we do perhaps best is to reconnect people with the joy of learning and teaching. As you'll see, I teach teachers to bring this content into their classrooms, and some of the most satisfying work we do is to see this reinvigoration of teachers and the excitement that they have to get back into the classroom to do this work. I think some of the secret that we have, or some of the success we have had, is in this connection of taking university work. I'm a faculty member in the Department of Biological Engineering here at MIT and have been teaching this content for a dozen years or so to my undergrads. We take the authentic research questions that we are asking in the department, not only ours, but synthetic biology labs around the world, and we connect those to the biotechnology ecosystem, the industry questions that are important. And we want to partner, and we have partnered with high school teachers to figure out how to take those questions and bring them into classrooms. Teachers are really very much the not only the underpinnings, but really the driver of so much of the work we do. Here's a photo taken in a summertime day, not too far from here, of some of the teachers in a teacher professional development workshop that I ran, and some of the things that those teachers have gone on to do, because as I say, we really do try to enable the kind of work that people are excited about once they get some of the foundational work of the BioBuilder curriculum. So some of the teachers are writing, for instance, a Wikipedia page, one teacher's doing a board of directors for the nonprofit, one wants to generate a science Olympiad event using synthetic biology. One of the teachers has taken the high school curriculum that I wrote and adapted it for middle schools. We've had presentations at national meetings, both scientific and teacher professional meetings. Some teachers like to do public outreach events to engage PTAs and other communities with science and engineering. And some of the teachers have extended the protocols to better suit the kinds of things, the reagents and the materials that they have in their classrooms and labs. So we really are a malleable and adaptable curriculum that's driven by teachers. We are also an open access content. So all of the content we have is provided online. This is the homepage for biobuilder.org. And the content, the curriculum is available as modules at BioBuilder. We have also collected it over the summer, and it's been published in a book that O'Reilly has published as of July. Each of the chapters is really downloadable on the website, but if you like the book, which I like to have a book in hand, it is available through O'Reilly and Amazon and other outlets. So let me show you just one of the modules that were part of the module, some of the resources that we have as part of the content for BioBuilder. This is one of the labs that we run in which bacteria have been re-engineered to smell sweet. So they smell like bananas rather than the normal stink that bacteria have associated with them. And this lab has been run just over the last couple months in Tyngsboro, Massachusetts. Somebody sent me a photo of the students doing the work in high school in Tyngsboro. Another teacher sent me photos of his students doing the work in Taipei, Taiwan. And they're both doing the same, they're all doing the same lab. It's happening around the world. And the students are working to understand how two competing genetic designs might behave if the specification for those designs could be satisfied with either one. And so the students are pursuing a authentic research questions in labs around the world. This is what the teachers portal for this experiment looks like and some of the resources that are available. You see on the left, there's a comic strip which can be used to set up the content with students. And those comics have links to further animations between the characters in the comics and also to the teachers' preparation videos for how to get the lab materials ready so that their students can carry out the investigation. The website also has freely downloadable posters that are descriptive of the kinds of experimental questions and the kinds of techniques that you would engage in if you were to carry out this experiment. And the reagents themselves, the lab materials, have been packaged and are distributed through Ward Science. Now, they are a for-profit company. I'm a public benefit company, so they have a price point that I have no regulation over whatsoever. But the materials are available and to be ordered into education. This is a picture of a quick guide on the left and a manual that Norman here has helped to design and publish with me, which is what is given, again, on the website and comes with the kits as well, to teach students and really anybody how to carry out these experiments. And there's one for each of these labs that's available on the website. So that's an example of the content. Other content that we have relates to other authentic questions that we're asking in research labs, things like how do you choose which cell type to run a genetic program in, right? Not all cells are created equal. How do you model a system? How do you rationally combine different genetic elements so that you can predict the outcome of a circuit? And these are questions that folks in the field don't know the answers to, and we have made them into modular units that can be carried out in labs in high schools clearly around the world. Some of the future of BioBuilder includes some exciting things. Some of them are further professional development workshops for teachers. I have really enjoyed teaching teachers. They are wonderful learners and they know how hard it is to teach. And so it's been a real pleasure to work with teachers and they become ambassadors for the program. So if you know of a great teacher, if you think of a teacher that you might believe is would really grab onto this kind of content, please send them my way. We run workshops all over the country. Workshops are always co-taught with a faculty member and a high school teacher and last usually on the order of 3 days in the summer. We also run an after-school club called BioBuilder Club. It is actually very hard for teachers to bring new content into their classrooms, high school teachers especially. There are many frameworks, many points of resistance that they meet. And some of those include a confidence that they may not have in teaching, especially the engineering associated with synthetic biology. And so we have been building up the BioBuilder Club over the last few years. It runs quite similarly to iGEM, for folks who are familiar with iGEM, except that this runs during the academic year. We have a kickoff event in October and a concluding final assembly in March. And we have check-ins every month with the teams and online group meetings so that we give them sort of ongoing support for their projects. We also celebrate all levels of design as well as completed testing, building and testing of circuits. So there are teams, there are schools that don't have the capacity to do the laboratory work, and so we help them understand what it is they're trying to design, help them troubleshoot and give practical ideas about if they were to get into the lab, how they would get started on the lab. And so that is running. We have a final assembly in March, March 17th down at Lab Central. And so if you would be interested in hearing the students present, we have teams all over the world. So some will be Skyping in, many are from New England and will be coming in. The Chicago team is flying in, and the students will present their work to each other and to the community. We also have, as I mentioned, teachers who are adapting the content so that it is more suitable for middle school. I think a lot of, I would say girls especially, but many students sort of self-select on science and engineering as early as the middle school years. So that would be 6th, 7th, 8th grade. And so some of the teachers are adapting the content that we've done to middle school and are collecting it on the in your site. It's still a fledgling project and plenty to do if you have interest in that area. Some of what we are dreaming about for 2016 includes a community space where we can bring the biobuilder content into practice sort of 24/7. Our plan is to dedicate a teaching lab inside of Lab Central, which is a biotech entrepreneurial community just down the street here in Kendall Square. They have 25 or so resident companies, small startups, that are accelerated in their growth within the context of Lab Central. It is a shared office space that includes laboratory space that they can rent. So the companies that are there can rent a bench or a private laboratory suite for up to 2 years to get off the ground. And these are all small, high impact and high potential companies. Lab Central is going to be expanding about a year from now, and when they expand their footprint, we are planning on dedicating a teaching lab inside of that entrepreneurial community. I think it's an exciting prospect because now, you can imagine a student coming in and being able to talk to an entrepreneur who is building out a company using these techniques that the students are learning. Teachers can continue their professional development throughout the year if there's a space for them to do it. So I think it's an exciting prospect for the neighborhood. I'm excited to be part of the ecosystem that will bring it out here now. So that is my story. If you have questions, my contact information is there, BioBuilder at Lab Central, info@biobuilder.org. All the BioBuilder stuff comes to me in my inbox, and I would be really delighted to talk with you. And if there are ways that you think you would like to volunteer, contribute, have some part in BioBuilder's growth and next step, I would be really delighted. Can I take a few questions already, or yes? Send them to me, and I reply. I will reach out to them and talk to them. I talked to a teacher in the class this morning by Skype, and at this point, it's not a strategy for building tremendous capacity very quickly, but at this stage, we have 400 teachers around the world really, who can help if any of these teachers are local to the Bible after this. Yep. No, it has grown quite organically to be in countries around the world. So I have in the BioBuilder Club a team in Shanghai and a team in Taipei, and then I have teams in Chile and Canada. The challenge in getting the materials to other countries is non-zero, right? And so shipping the kits themselves to those, so the company that is shipping those experimental kits, it's very slow, it's very expensive, it doesn't always work well to ship it out to those countries. But we try. And we try to support them in multiple ways. Yeah.
Because I was thinking for the kids to put access amazing features and facilities, maybe most of them are in the city. So I'm thinking how we can reach the kids also from the countryside. Interesting part is I was born in an area where the complete countryside.
Yeah.
But I do think actually I'm confident to see my relationship to nature and leaders really, really intimate because I have my own interpretation of
Yes.
all those microorganisms.
Yes.
But I think now science coming to give me a new interpretation. It can be a very interesting kind of actually co-learning and co-teaching.
Yep. I think it's brilliant. Did everybody hear that? I think the notion is biology is all around us, and some of what we can do is deploy or use that surrounding biology to really change the way we think and change the way we learn. And there is nothing that says it has to be in Kendall Square, right? And I think it's a very interesting prospect to bring it to many settings. It's harder to adopt it in some settings than others. And so I've been at this for 5 years, and I will say that it is the better resource schools, the ones that are more flexible timing and testing and things like that that find an easier way to get this in. But I am very interested in bringing it to whoever would want to do it. You know, that is really my goal. Is any teacher, any student who wants to do this should have access. Yes, ma'am. Yeah, so the website is fully open. All the animations, all the instructions, all the chapters from the book, and all the all the support information that I can put up there is all open access educational resources. The physical material, the DNA and the bacteria, when the company sends them, gets sent to schools. It could be sent to a community lab or things like that, but you need biosafety level, biosafety oversight to receive those kits. You have to check a box that says that are you compliant. Right. Well, contact me if you have if you really would like to. Okay, thank you. Thank you very much. Can everyone hear me all right? There's also a speaker.
Yeah, so first of all, I just wanted to thank the organizers of BioChanges. I'm very excited to be here and to talk about Boston Open Science Laboratory. And my name is Angela Kazmarzik, and I am one of the co-organizers at Boston. And we are a DIY bio community lab space for the people of Boston. So right now is a very exciting time for molecular biology, and 2016 has proven to be a very exciting time for all of us at Boston. So we've been working really hard to reactivate the space, and recently, just this past week, we officially became incorporated as a nonprofit organization. So we're really excited about that, and you can find our lab space on the second floor of the house in the middle of the neighborhood that's only about a 5-minute walk away from Davis Square. And we're just really excited to be a part of this growing DIY biology movement. And this is only the beginning for us. So who is DIY biology for? So I think the simplest answer to that is everyone and anyone who is interested. So one of the major missions of DIY biology is to make biotechnology accessible to the community and to give people the opportunity to do molecular biology in a laboratory setting. And we want to do all of that at Boston. And we also want to foster collaboration between people of diverse backgrounds and expertise to do and learn molecular biology in the lab. Or, as we like to call it at Boss Lab, molecular hacking. So at Boss Lab, we envision building a biohacking community that consists of several different groups. We want to include citizen scientists of all ages, whether that they be young aspiring scientists or retired individuals who are looking for a new and exciting hobby, such as molecular biology. And we also want to get students from local high schools and community colleges and even at the university level involved. And we also want to give academics and industry professionals in biotech the opportunity to give back to the community by either giving a talk about their science or teaching their science.
So right now, Boss Lab is working really hard to build your DIY bio community right here in Boston. So why DIY biology? So can I get a show of hands? How many of you are very familiar with the DIY biology movement or biohacking, garage biology? Okay. So a good amount of you. For those of you who are new to it, this term DIY bio and this movement was actually born right here in Boston. So it was back in 2008, a while ago. That was actually right before I started grad school. Matt Powell and his colleague started a nonprofit organization known as DIY Bio. And they created a website called DIYbio.org, which was intended to be an online community and forum for biohackers around the world. And since then, it's really grown. And so one of the major motivations for this movement is simply that molecular biology can be very expensive with all the reagents and the equipment that we use. And so one of the primary goals initially was to reduce the cost and of doing molecular biology and to make it more affordable. So a great example of this was the Open PCR project. And so you see to the very left over there, it is the Open PCR project was initiated by Peter Jankowski and his collaborator. And what they did is they built an open sourced protocol or a blueprint to basically build a very affordable thermocycler so that anybody could do PCR. DIY biology is also has a lot to do with citizen science. The idea of bringing the public and engaging the public and getting involved in scientific research. And since 2008, many DIY biospaces, including Boss Lab, have popped up all around the world. And some very exciting projects have come out of these spaces. So for example, a few hours from here in Brooklyn is GenSpace. It's another DIY biospace. And they worked on several projects, and one really interesting one was the Urban Barcode project, where they got local high school students involved and taught them how to use DNA barcoding technology to basically test what are the actual ingredients in their food. So is that steak, is that meat from a cow or from a horse? And some really interesting molecular biology and synthetic biology projects that come out of these spaces. So for example, at Caniculture Labs in Oakland, California, they started a project called Open Insulin, which is a project to develop an open source protocol for engineering E. coli to reduce insulin in hopes of eventually reducing the cost of insulin. And we also have a really interesting, fun project called the Real Vegan Cheese Project, which was a collaboration between BioChanges and Caniculture Labs on the West Coast in California. And what they did is they basically worked on engineering yeast to produce cheese proteins so that eventually you could make cheese that did not require any cow's milk. And so this movement has really grown since it really first started. And these are just a few of the examples that have come out of this movement. So for those of you who are concerned about safety, back in 2011, the DIY Bio Community and the Woodrow Wilson Center in Washington, D.C. got together for some workshops to basically develop codes of conduct for all DIY bio spaces. And they also partnered up with American Biological Safety Association to produce a page on DIYbio.org where people could ask a biosafety expert basically anything on how to conduct molecular biology safely and how to dispose of waste properly. And many of these DIY biospaces also have formed these science advisory teams. And we actually just formed one here at Boss Lab. And the idea is basically to review project proposals by members to determine whether or not it is safe to proceed with that project in a DIY bio lab setting. And Also, another thing is that Boss Lab, we've been working with, thanks to Natalie, we've been working with a environmental and health safety specialist to help us basically make sure that we're doing all of our science safely at Boss Lab. And I'd just like to also add that there have been a few surveys that have been conducted on these DIY biospaces around the world. And it seems clear that the whole community is very serious and proactive about safety. And I think that the benefits of this movement greatly outweigh the potential hazards of it. And here at Boss Lab, we're very serious about doing fun and safe science. So we've made a lot of progress here at Boss Lab. So we've been working hard to make it more active and engaging the community. And this is really a new beginning for Boss Lab. So this past summer, we developed a new team, and we started off by holding a few journal clubs, and then we held our own lab course, a molecular biology lab course. And after that, we even started a few new and exciting projects in the lab space. So there's a lot of things to do at Boss Lab, and we think that there's something for everyone to do here at Boss Lab. And so this is a quote that I really like from a speech that Meredith Patterson made, who is a fellow biohacker on the West Coast. And it goes like this: scientific literacy is not science education. A person educated in science can understand science. However, a scientifically literate person can do science. And so this is something that we're really striving for at Boss Lab. We don't want to just allow people to vaguely understand biology. We want to promote scientific literacy. And that's very important to us. And we want to give them the tools to be able to do the science in a laboratory setting and to understand what they are doing at the molecular level. And so some examples of this is that we've started a bi-weekly journal club where we can discuss and learn how hot new technologies like CRISPR-Cas9 work and how you can apply them. Also, there's a nice photo in the middle of Dennis, who is one of our other co-organizers. He has been running a recurring study group for the online edX synthetic biology course. And we've also been regularly holding project nights, which are great for newbies and people who are curious about Boss Lab to actually come to our lab space and check it out, and to share ideas and hear about new and exciting projects that are currently happening in Boss Lab. And so as I mentioned before, we held a molecular biology 101 lab course, and this was really exciting. We had 5 students, they were 5 amazing students, and they consisted of, some had a biology background, and others had no biology background at all. Some were software engineers. And we even had a PhD in chemistry come to their class. And so for the class, we did bacterial transformation. So we introduced GFP, green fluorescent protein, into E. coli, and what we ended up with were green glowing bacteria. And so we're excited to hold more classes at Boss Lab. And for example, we want to hold a synthetic biology lab course where people can do real synthetic biology at Boss Lab. So to design, build, and test, and repeat, so that we can build genetic circuits in these microbes. And we're very excited to have more of these courses. But we'd also be very interested in getting some more grad students and PhDs involved in teaching some of these classes. And so now on to the projects. So we have a couple of really exciting projects that we recently started that we are initiating at Boss Lab. And so one of them is the Truffle Project, which Raphael, who is a co-organizer at Boss Lab, has started. And the idea is to isolate and identify microbes that are living with the truffles and to find the microbes that produce truffle aroma particles. And we also have a very exciting proof of concept project that Matt in the lab is working on. And the idea is to engineer yogurt bacteria to produce NAD+, which is important for metabolism, but has also been shown to induce longevity in mice when they're infected. And we also have some very exciting ongoing projects, such as the microscopy hack project that has been run by Will and Ed in the lab. And one thing that they've done so far is they've built a automated stage so that you can do automated microscopy. And so if any of these projects have sparked your interest, please come and find me or any of these other amazing people on our team. And so, yeah, these are the co-organizers, the current co-organizers at Boss Lab. And it's been a lot of fun, and it's been a pleasure to work with all of them. And we consist of software engineers, PhDs in biology, and designers. And yeah, we're excited to be a part of this movement. And you can check us out on our website. And you can also check out our meetup, which lists all of our upcoming events. And just a quick announcement, our next journal club is going to be on February 9th. And Raphael will be talking about, presenting a paper that is related to the truffle project. And thank you very much for listening.
I think we'll just have one more talk and then we'll take a 5-minute break so we can stop it and then we'll get this with this team.
So we're back on stage to introduce this project called Biologic. This is the whole team. So Jufe and I are going to represent the team and introduce this work to you guys. We've been working on it for the past 1 year, and now it's almost at the end of it, and we're working more on newer technologies now. So when people talk about synthetic biology and biological engineering, it sounds like a very new technology. But indeed, we try to actually find the historical stories out of bacteria. If you think about it, it is actually one type of life form that has been existed for the longest time on Earth. Most of the affairs people had with bacteria came from the food. The bacteria exist in your wine and in your pickles and in kimchi. So this bacteria we are looking into for the biologic project came from the historical culture of Japan. It's called Bacillus subtilis natto. It was found the first time within the dry rice stalk. A Japanese samurai found it during the war, and he by chance found the soybean that were carried in a dry rice stalk made bag tasted so much better, and he looked into why the soybean changed in his taste and figured that was due to the bacteria inside the dry rice stalk. It was named natto, but in Chinese and Japanese character, it literally read as dry rice stalk bacteria. And this is how the bacteria look like. They're basically this raw-faced bacteria. And for the biologic project, we're trying to find a new function of this Asian bacteria. that is its own hygromorphic transformational behavior. Basically, it means when we modify the relative humidity inside the environment, we can also trigger the shape and the size change of this bacteria. It expands when the relative humidity in the environment is higher, and gonna contract when the relative humidity is lowering down. And so we are able to use this bacteria as a, let's say, nanoactivators. We made a hybrid biofilm. We're basically layering this bacteria on top of an inert film. In this case, it's just a stretchy latex film, and make this bilayer hybrid film. And as we modulate the environmental relative humidity, this film can bend and unbend itself. It becomes almost a bending actuator. And this is a diagram to show how it works. Each bacteria expand and contract by itself. And when you align them one by one, they connect with each other and form a biofilm, a bacterial film. And you put this bacterial film on top of another inert film. This tool, when the relative humidity changes, because they have differential expanding rate, because this entire sheet material bends up and down. And this is a real sample that we are able to produce. As you see, when we lower the relative humidity in the environment, it curls up by itself. And the first thing we did is we are trying to figure out how to culture this bacteria in large quantity in a biolab, and also study the transformation behavior of this bacteria. We just use the very standard cell culture process to culture this bacteria. One bacteria can grow into millions and billions overnight. And this is an illustration of the whole process. Again, it's standard, and incubating and purifying it, suspending it into water to make it into the right concentration we want. And this is an experiment we did when we put this bacteria film into a dark light microscope and use a straw to blow in different air with different relative humidity concentration. You're able to see the bacteria is kind of getting smaller and bigger, so it's changing in real time. And based on our measurements, we noticed there is over averagely over 11% elongation along the longest axis of this bacterium. So basically, it grows 10% more when the relative humidity changes from 0% to 100%. And we think this is due to the majority of the performance is due to the protein contents within the bacteria. The protein are very good at absorbing and dissolving water and try to keep a balance between itself and also the environment around it.
So we talk about like basically the whole talk gonna separate into 4 scales. So we talk about the first scale at the micro and nano scale, which is about the culturing the cell. So basically, then the next step is we are looking at how can we digitally fabricate the cell. So this is a illustration of how we start using the digital fabrication to composite the cell with the latex. So the first is, of course, the cell is mixed with the water, and then you have this liquid-based solution actuator that you can actually deposit using a bioprinter. And then so when you print it on this flat surface, because they're all swelled, and then so basically they're all like basically full of water, and then when the water evaporated, they will start to shrink, and then because they are attached on the surface so firm, so when they all start to dry out, they will basically pull up the whole film and to bend. So you can see that basically as you're changing the relative humidity from 30% to 90%, and you can basically make this bilayer structure releasing and also bending, and then creating this kind of an actuator. And then we actually studied basically trying to model how this transformation is related to how much amount of cell you put it on it. And since we can measure the force basically when one single cell is shrinking and swelling, or how much force you created, we can basically simulate when you print certain amount of cell on the substrate, how much bending angle and bending force you can actually create. So with that kind of knowledge, we are able to create this software simulation platform where basically you can specify how much cell you can print on that. And then if you're basically having a three-dimensional model, and then you can say, specify, hey, where I want to put this cell, and then you can basically simulate what kind of transformation you would have. And then with this simulation, if you think, oh, this looks nice, and then you can really go into the next step of really think about how can you fabricate this transformation. And then we're thinking about three types of fabrication methods. The first one is, of course, manual pipetting. That's actually how we started. So basically, we spend nights and nights like trying to perfectly pipette the cell solution on the substrate, and then it's kind of like sometimes you get nice results, sometimes you see still it's manual pipetting, so it's not that perfect. And then we start to think about how can we use the machine robot to control that more precisely. And then so the first platform we're developing is the inkjet printing platform. And so this is a quick video showing that basically we construct this small build platform, and basically inside you have this syringe where you can extrude the cell. And then after you printed that, and you basically, so basically this is showing the process where you have a software platform indicate the printing path, and then the printer will print it for you. And then let me go back a little bit. And so after you print that, of course, you have some process of cleaning the printing head and then drying the substrate, so you get the final result you have. That will be, so basically cell loading, dispensing, and then printing, and then cleaning the cartridge again. So basically this is the process we're thinking about, like how can we print the cell. And of course we are also trying to model like basically how much amount of the cell you can print in relation to the feeding rate, and also the speed of the print, the print actually moving along the x and y axis. And so this platform, we basically wrote a paper and published recently in the new journal called 3DP plus three-dimensional printing additive manufacturing journal. That's chief editor is Stella Tutti from architecture department. And if you want to look into more detail, you're welcome to reference in this paper, in this journal. And furthermore, we're starting to think about, since we already have this printing platform, how can we go beyond printing just the cell? Can we print something more? And then we started developing this modularized printing system where we can basically attach different printing modules and also other accessories such as fan, heating element, or UV light, those elements for more other functional or smart material printing. And this is like basically a short overview of different components you can actually attach on this platform. And then we're also further developing this software, simulation software platform where we can basically specify area, line, and also filling method that we can use for different type of material printing. And those are showing different type of material we can print that. Metal cell is one, and then we can also print the arginine solution together with calcium chloride. And also the third one is printing the hydrogel and using the UV light to cure it. And then the third one is for like artists to print the thermochromic ink. So basically you can print something more than just the passive black or colorful ink. And then beyond that, recently we start to think about how can we basically make a much larger printing system where basically we can, later we're going to introduce one of our applications is to print the cell on the fabric. So basically, but before we have this small scale printer, and then basically the fabrication process is still be difficult if we want to make something a little bit large. That's why we start to constructing this large scale printer where you basically can reach to almost 1 meter by 1 meter print area and has the accuracy at 10 micron. And this is done by together with Yannick Peters, who's a VT student in our group. And so basically, once we have that, we can go start looking at the third level, what kind of transformation primitive we can build. So we basically summarize it. There are two types of primitive. One is curved bending. One is angular bending. For the curved, is basically you have a soft substrate, and then you evenly distribute the cell on it, and it will bend to form a curve. And then for the angular, is that you basically print the cell in one line, and then you basically reinforce the two side of the substrate, so you can really bend. You can basically get a sort of like a similar to origami folding structure. And then with that, you can get one-dimensional linear transformation. So basically, on the left, you can say the rendering is a simulation, and then the red one, the yellow film is the actual performance once we print it. And then we can also create a two-dimensional expansion and contraction, and through that, we can basically create this kind of radius geometry that we see expand and close. So basically, the whole idea is that how can you expand beyond just a single film bending and releasing? How can you use that to create more various transformable primitives? And then we can also create a 2.5D texture change, where basically you deposit the cell in a certain pattern, and then as the humidity change, you can have some like texture appearing and disappearing like when you touch it. And then for the last one will be basically you create a three-dimensional origami structure where you can deposit the cell at the hinges, and then as the humidity changes, you can basically create this kind of folding and unfolding effect from 2-dimension to 3-dimension. So that's pretty much about the transformation primitives, and I think.
Yes, so what can we do with this? The first thing we came out with a lampshade. With the origami primitive Jiffy just showed you, we designed this lamp. We basically just patched almost 100 little transformable units on top of a 3D printed skeleton. And here, as you see, when we turn on the light, the heat generated by the light causes the relative humidity to decrease around the cell, and the cell shrink causes the unit to shrink, causes the lighting pattern getting out of the lampshade to change. Another application we're looking into is how you can use this material to augment a natural organism. And here you see when we print this bacteria along the natural vein, the silicone film starts to behave like a real leaf. When we think about nature, we can't control it, transform based on season. But here we are trying to look into this artistic idea of trying to be able to control the behavior of nature, to let it better interact with you. In this case, a tea leaf can open up as you are pouring tea, and as your tea is ready, it seems like the tea now can actually become alive and start to talk to you. Or a flower that can blossom with both shape and color when you water it. So in this case, we actually mix the bacteria with the color-changing chemical pigments and also the liquid latex. So the final film that changes both shape and color is produced by this mixed composite liquid material. Another thing you can do is you can now also print in conductive traces on top of the biofilm. By the end, you've got this film that can be controlled by the Joule heating induced by the circuits. And this is a toy application we are able to make with this Joule heating induced relative humidity change idea. As you are folding and then unfolding the corner of the paper, you are turning on and off a heating circuit. And the heat generated can actuate this paper toy. Some toy companies in the US, such as I think Hallmark, yeah. So they really like this idea because they told us kids like electronic kids. However, we don't want to put electronics into toys that are for really young kids because it's kind of dangerous to put it in their mouth or whatsoever. So in this case, it's almost electronic free, but they are animated toys. And so by the end, the final application we decided to focus on is the wearable, the second skin idea that we are able to achieve with this material. Surprisingly, it's super sensitive to the tiny relative humidity changes. in this case, the bit of temperature and humidity changes on the skin can cause the transformation of the film. So we reached New Balance. It's a long-term collaborator from the industry, and they told us a lot of knowledge in terms of how people, how the heat and sweat get distributed on top of on the surface of human body, on the skin of human body. But then we are putting those data into a parametric design environment and started to look carefully into the heat and also the sweat and designed our parametric kind of a pattern based on the heat map. The idea is the part that sweat the most will have the densest and also biggest opening of the flaps. And then we put it into almost a production process. This is the whole team that worked behind the final garment, which I'm gonna show for on the next slides. It includes biologists. That's why I think she's here. Also, we invited a professional fashion designer coming all the way from London, and she helped us to sew everything together. By the end, there's also Chini, this parametric designer, helped us to design the whole pattern. And this is the final fabric we're able to make. So again, it's a composite between the normal fabric and also the responsive film. It reacts to the sweat, and once you heat up and your body sweats a lot, it starts to open up to help your sweat to get vaporized much faster. And this is the final suits we created. And this is the final video we would like to share with you guys. So just enjoy it. You make it as an art. It's about 3 minutes. So the two dancers featured are both currently active dancers over Boston Ballet. Here, for the ending part, we're just trying to depict the fact that this bacteria doesn't only exist in a cliche form that are on stage, but also exists in the real life. Thank you, everybody. So I think we'll start with our
Yeah, we'll just take a 2-minute break while we're setting up the projectors for Joe's presentation. So if anyone needs to use the
yeah,
bathroom.
please stay. So this is actually the most amazing
We don't need to move. Yes. Put it on the table.
by the way.
So you're you want to center? Need the second. Oh, you got it. Yeah. Oh, food on. We're gonna have to split the screen. Somewhere like right. Yeah.
Probably want them back here as far as you can. Yeah.
Well, it's gonna have to be small for both of them.
Yeah, but still, maybe this. Well, we have to see.
Yeah.
All right.
Yeah.
Please grab some cheese.
Yes, yes. You have to get used to it.
Joe, do you have the other? The top. Have you seen my?
Is this the other computer?
No, the other computer will be yours. I'm just trying to. Trying to fit. We'll figure that out in a minute. There you go. You can talk to everyone who's in London.
Dark.
But if you change the slides,
When the slides come on there.
Yeah, I know. I want to find something.
also it's automatic.
Don't you have something? Oh, here.
So go check it.
Cheese.
Cheese, no.
No.
No cheese.
Or fruit.
I'm on diet.
Good job. I'm supposed to gain it.
By the way, that's the typing condition.
Yeah, that's the kind.
Okay.
That's why I get it. Practice.
Okay. But you add some information like 3D review.
Yeah, because I wanna.
See?
Yeah, but there's something.
Wow. That's so cool. Wow. Artist work. It's very different. So why they need two projectors? Oh really?
Hold on.
Yeah, have them to adjust.
Can you help on this?
I just need to get focused. No, it's the same. They're kind of different ratios. It could be.
Your choice.
Let's just give people time.
Is it good? Or something's missing? Oh, yeah, another.
Good to go? So I guess thanks again for everyone for coming. And in terms of biodesign and bioart, the person that's about to speak is possibly the person who's probably most world renowned for doing this. Biodesign is kind of a, there's a lot of hype around it at the moment, but Joe Davis has been doing this since 1985, 86 maybe. And the idea is that he's going to give his talk on what is, it's a mix, I guess, of his current work and past work. Yeah, and so it's people like Joe Davis, like Boron Katz, and a few others that have been doing this for so long, and that's the idea of, I guess, around BioChanges is, one, getting the communities together, having these events and trying to build facilities, but also recognizing the people that have done it, before we ever started to even attempt to do it. So I'll just hand the floor to Joe and take over.
I'm really not going to talk about what I've done in the past. There are a couple of slides about some past projects, but they're only there for continuity. This is all about what I'm currently engaged in. These are flowers that we grow in space recently on the space station. You'll see there's a place with this. But I'm going to talk about a very, very unusual, very odd sort of garden, not the kind you'd imagine. This garden appeared about 4.5 billion years ago, the first time. And as far as we know, it was the early Archean, roughly about 3.5 billion years ago, that life left its first detectable impressions. These consist of bacterial microfossils. It's a time when this crust had just cooled enough so that continents and kind of plates to form. The atmosphere, well, for most of the history of life, the garden had a very different atmosphere than the one we know. There was no oxygen. It consisted of CO2, ammonia, sulfur dioxide, methane, and hydrogen. The organism is thought to have evolved in this reducing atmosphere. Can you all hear me? I'm doing it good, right? So, even though they evolved in this poisonous atmosphere, because that atmosphere would be poisonous to all air-breathing creatures today, the action of those organisms in this poisonous envelope of gases changed it into an oxidizing and oxygenated atmosphere after billions and billions of years, hundreds of billions of years. Yeah, you can see this oxygen only appeared in the west curve of this long spiral that you expect. The most widely accepted chronology considers the Great Oxygenation Event, the GOE, which occurred sometime about 2.5 billion years ago. And the organisms responsible for changing the atmosphere into an oxygenated atmosphere actually appeared several hundred million years before the oxygenation event. Cyanobacteria thought to have significantly contributed to the Great Oxygenation Event, to the change in atmosphere. Didn't seem to have appeared until about 200 million years ago. It's interesting too that oxygen in atmosphere was created as the waste product of these original organisms. Kind of the reverse, where we're filling the atmosphere now with CO2 as our waste product. But until about 0.85 billion years ago, the level of oxygen in the atmosphere was nil. It was 1 to 2% of for most of the time of the level of oxygen today. And this 1 to 2% was enough. It was it was the causal agent of the most devastating biological disaster that ever hit the planet. Because oxygen is toxic to obligate anaerobic organisms. All of the breathers of the previous atmosphere were doing this fast. And it's interesting that current multicellular life isn't the first time multicellular life appeared. Apparently it's appeared at least 20 times before. These extincts, these are fossils of extinct multicellular organisms that lived within the 200 million years within a few million years of the oxidation catastrophe. And oxygen in our atmosphere reached its peak at 35% sometime in the Cretaceous, only to spike down again at the KP extinction. So I'm an avid fossil collector. I have a big personal collection. I have this fascination with fossils. This is a half-billion-year-old cephalopod. I found a stone this big that's sitting across the street in Thomas Schwartz's lab. They grew to like 20 feet long, longer, these pre-cephalopods. Anyway, why am I fascinated with fossils? It's because they have this ability to transport imagination, to bring you into lost worlds. And maybe for that reason, there's also something terribly plaintive and poignant about fossils. Because they also represent the great bottlenecks in history when life simplified. It was the passage of, yes, genetically diverse, genetically simplified populations. And the great oxygenation event wasn't the only mass extinction. It was preceded by many others. In the Ordovician, 96% of everything died. And then in the Devonian, 70%. Then in the Cretaceous, more than 70%. It's just each time life, the diversity of organisms. There was more diversity historically in Ordovician 500 million years ago than there is now, and Earth never recovered its the diversity of its biome until almost recovering it in the Cretaceous, and then there was the KP extinction. Yeah. We lament the passage of knowledge, the beacon of learning at Al-Andalus, the Library of Alexandria in Egypt, or the Library of Nalanda in India. Each one of these very tragic losses of all the treasures of culture and civilization reaching back thousands of years. All the book burnings and acts of iconoclasm, and well, all these are kind of trivial by comparison with the unspeakable biological disaster of the mass extinctions. There are hardly any numbers to describe the extent of the lost libraries of genes and proteins held in uncountable disappeared species and whole kingdoms of life. More than 99% of all species that ever existed on Earth are estimated to be extinct. I mean, we can only imagine what fields like synthetic biology could accomplish with this like vast retinue of lost genes, even if a tiny percentage of them were recovered. Unfortunately, we don't have much left in the toolbox. Most life on Earth lives in saline environments, in oceans and seas, both on the continental shelves and in the benthic deeps, in deep hypersaline anoxic basins, and in deep ocean banks. Terminal hypersaline environments, evaporating seawater ponds, lagoons, marine saltpans, solar saltpans, and the great salt lakes of the world. The Dead Sea, Great Salt Lake, but there are many, many others. This one's caribou gases on the Caspian, and this one you get from Africa, etc., etc. There are many, many salt lakes in the world, and also very high temperature hot springs environments, hypersaline hot springs environments. So, as it turns out, human beings and other air-breathing, freshwater dependent organisms, why we're by far the latecomers. We are the extremophiles. It's like our self-centeredness kind of skews what we think of as extreme. So halophiles are extremely diverse. They can endure extreme temperatures and extreme humidity. Consist, yes, in environments without any water or very little water. Routine exposure to high UV at high altitudes. There's a deep lake, it's called deep lake. It's a hypersaline lake in Antarctica, in the western hills of Antarctica. The temperatures range from 11 degrees C to minus 20 degrees C. It never freezes because it's so extremely saline. And it's harvested various species of halophytic organisms. Here's a map. This is a map of all of the subglacial lakes. Liquid lakes also have to be hypersaline in Antarctica. This is one that they tried to drill into. They stopped short, 330 meters. The last season, they never made it all the way there, but they will. Some halophiles are known to survive in high vacuum, radiation, and temperature extremes of the space environment. The long duration exposure facility that NASA flew in the 80s and exposed leads in this century. There's actually several of those. There's an exposed E and an exposed R flights that went to the International Space Station, and the organisms were exposed to, say, radiation in space environments. And these are European experiments, the European Technology Exposure Facility, also on the space station, and BioChanges, which were lofted by Soyuz rockets into orbit and with exposed and then deorbited with thermal blanket, and some 6 of those experiments are unfolding. So I do have an interest in space. I've been interested in myself. I need to know who I am, where I am, what I am. I mean, that's the measure of sanity, right? You have to know these things. So I do projects to transmit messages for extraterrestrial intelligence. Ironically, much more about transmitting messages to ourselves. Nevertheless, these are the most powerful transmissions to sunlight stars ever conducted. The first one at 100 million watts for 20, 30 minutes. The second one, several hours at 2 million watts from Arecibo, Puerto Rico, in which we transmitted the gene for Rubisco, the most abundant protein on Earth. The protein that it's not very efficient, but it takes inorganic carbon from atmosphere and turns it into food. We don't make Rubisco, we human beings, but all of the complex carbon that we're made from, that we consist of, was produced at one time or another by this protein. There's a collaborator in the Rubisco stars project. But these are the things, this notion about the failures of radar transmission, sort of shortcomings of radar transmission. subject to inverse square law, so the B is you're going to get the column with the distance, and you can send one signal if you can manage it. There are only a few radars on Earth bright enough to outshine the sun, so it's highly competitive access. And so you send one message for 3 minutes. Frank Drake's famous message from Arecibo was transmitted for 1 minute in 1974. So sun-like stars 25,000 light years away. Anyway, gotta survive. Radiation temperature, space environment, vacuum, you gotta have, because the speed of light is still a slow boat in the context of our own galaxy. It's 200 million light years across. So if we sent the message, if mitochondria Eve, our common ancestor, had managed to send a message to the edge of our own galaxy, and it was received, understood, and immediately replied to, that message wouldn't be back yet. And when it did arrive back, it would be different species, right? So bacteria, see, answer. I got into like coding all this different kinds of information in bacteria and made up these elegant, very clever strategies to keep mathematical parity with input data, but to be biochemically and biologically friendly with the organism. Activity I'm still engaged with, but see, I wanted to make biological spacecraft. That was the whole idea, because I could make enough of them overnight so that there would be one for every star in the sky. One problem with this is radiation resistance, because space is a pretty harsh radiation environment. There's cosmic and galactic sources as well as the sun. And radiation damages DNA extremely. Heavy ions, gamma rays, and X-rays all have their effects and blow up DNA. DNA organisms have these swing shifts, repair shops that go into action when damage occurs, and depending on the efficiency of that repair shop. See, we need radiation resistance to explore the planets, because the gravity well, we'll have to supply the planetary exploration with factors in space, and those will be synthetic biology factors, obviously, and these organisms will have to be radiation resistant. So I went to the nuclear reactor like in the 90s, in January 1994. I went to the nuclear reactor at MIT, and I had to bring my own incubator because the reactor primary cooling is 50 degrees C. It had to be pre-contaminated. So I thought it was pre-contaminated with tritium because once I put reactor cooling in the incubator, it was going to become nuclear waste. And so I had to find one. They didn't actually guard it across campus on a fern tree dolly. With my friend now, and now Professor Stephen Woodward at University of Idaho, we Japanese from 35 millimeter slides that is fogged from the radiation environment in the reactor, and the primary coolant was much hotter than it is now because it was actually fuel particles in the water. And, we made standard microbiological assays, over 20 different media for microtoxin, UVC, Terabacter, Bacillus, and etc. And we actually got growth in two of these media, a media called TCNZ for micrococcus, and a media called PYC, which is for yeast. And although we didn't get micrococcus disease, we got a strange thing. We had a species of Achromobacter predominated with numerous bacillus species, 5 bacillus species, and they're growing rapidly. And they were still radioactive, so you had to go in and re-inoculate the fresh BDM, re-inoculate until it was radiologically, but not biologically dead. You move it from the reactor, and then Boston University Hospital, Boston University, Hospital Clinical Microbiology Laboratory helped us identify the radios. See, that was a long time ago, and you can only grow this many organisms that exist. You can only grow this many. The rest is called unculturable. And we didn't have, back in the 1994, we didn't have many tools. You could only detect what we could grow. And so now we have something called 16S PCR, which is PCR is a. Do I have to explain to anybody what PCR is? It's a copy machine. See, to explain PCR adequately, you have to explain the structure of DNA. You know, the temperature, hydrogen bonds versus covalent bonds, and primers, and it's a copy machine. And you can select what part of the genome of anything you can search for particular things. 16S searches for genes that make one of the RNAs that comprise the ribosome. And this is conserved throughout nature with tiny differences here, enough to be pretty specific about what family of organisms. And so these are my colleagues, some of them in the room who reacted to Project House, decided to go revisit these with contemporary tools, sort of, and using 0.2 micron filters, Connor, Hampton, and Cobalt, tested them for us, and notified the reactor about what our volume and rate. We're going to use 5 liters of reactor fluid. We had to get a 50-degree incubator because we're going to simultaneously do microbiology. We're going to grow what we can grow, and then we're going to look with metagenomics about what we can find. And Michael Napolitano is like he's a brilliant member of our lab at Harvard. And one of the cool things he's done is to collect this like library of species of stuff that he's found growing all over the lab, like probable contaminants. Among them is the radiation-resistant organism Deinococcus radiolans. And so we have these like little pneumatic rabbits from the reactor that will let us put it in these tubes like Dr. Van Kelly inside. I have 4 everywhere, so I can put 4 samples and just will shoot into the reactor core where we get 10 to the 13th neutrons per square centimeter per second, and an unquantifiable, thus far unquantifiable bombardment of gamma rays. So we've accumulated. Yeah, we're going to use MySQL. After we do the after we do the RNA sequencing, this PCR to tell us which family. It's also going to do shotgun sequencing, deep sequencing, one sequencing, what it can find, and that's with this guy. And we're also going to use axolotls, generative organisms. They're remarkably regenerative. They chop them into little pieces and they'll recover. You can take away many of them if you organs, parts of the brain. You can make excisional and incisional wounds with scarlessly. And they've been exposed to X-rays and UV in experiments in the past, but never to particle radiation. And so, one of my collaborators is Ben Martin, back there, who's laboratory at Northeastern, who's got an Axel Ranch, and we're going to expose 20 or so. We're going to take pictures before and after, expose 20 or so axons to varying exposure to 10 to the night. Neutrons per square centimeter per second out of the beam tube. And we're going to do transcriptomics to look for spikes in activity of proteins quickly in response to radiation. And we're also going to use tissue from the standard laboratory mouse use, and from a practically spiny mouse, a newly investigated regenerative organism. They regenerate amazing well. And we do horrible things to them all the time. But 16S PCR and metagenomics contribute to biological spacecraft in other ways too, besides finding for radiation resistance. At the back, we'll turn to salt again, all the data files. The oldest DNA, until recently, the oldest record for cat DNA is 700,000 years from a horse ancestor, those in Congo. And George Church told me that I should note that it's pretty deep enough into like 100 base pair fragments. Also, we have sequenced the mammoth genome, of course, and we've got the cat DNA for tens of centuries to identify. But see, unless they're found in these or other unusual circumstances, like frozen in under, the chances of finding readable DNA is really rare. There's spontaneous processes, notably determination and cytogene deamination, that are at least comparable to cosmic ray damage. Cosmic rays is seen in my cloud chambers, which I'm not going to explain right now. The exception to all this lies hidden in ordinary crystals of salt. See, when salt forms in brine, the salt crystals themselves expand and contract at a different rate than the brine itself, and so it ends up with these inclusions, these fluid inclusions in the crystal, and this has become a tool for geology. Note that Morton table salt, the salt that's on the street outside, is likely Permian or Devonian from mines in New York and Pennsylvania. That the only current salt you get is sea salt, and that's loaded with bacteria. So these are the inclusions between Permian halides and then Devonian halides. There's a science that's traditional in geology. It's called the fluid inclusion microthermometry, and it uses these inclusions in salt crystals and other crystals, like calcite gypsum. You take the crystal and you put it on a special stage, which is like a calculated device, and the PCR fluid there's some precisely heats and cools the crystal. When you do that, this vapor bubble appears at low pressure. The crystal is expanding, contracting, and vapor bubble appears, and then you gradually heat up the crystal until the vapor bubble disappears, and that's the temperature at which the crystal form, and this gives us, so the crystal itself gives us a kind of recipe for the ocean that it came from, but also it gives us the temperature of the ocean. So there's a kind of geomicropaleontology that's been largely ignored. It was first claimed in the 1960s, in 1960 and 1963 respectively, that living organisms, that ancient fossil organisms were brought to life from these crystals. And this was considered controversial, that contamination issues were suspected. Yeah, they were controversial. So, but in the 1990s, things changed. Then we got PCR and the tools, the basic tools of genomics and metagenomics, and we found many organisms. These things from crystals of southern Atacama Desert and Halobacterium and Archaea. This is all done with 16S PCR. And these are from Permian Triassic salts in England, Germany, and Austria, from the marine, from the shallow ocean that once covered Western Europe. One of these, a Halococcus, was cultured from several various geographical locations. A species of algae is also found, this Uriella. We think that its byproducts may help keep these organisms alive as a carbon source in the tiny droplet of saline, and also glycerol, one of its components, which would be probably metabolized by these organisms. And they shrink and they form these cyst-like nodules that were first discovered in these living organisms. And so decontamination is normally accomplished with sodium hydroxide, hydrochloric acid, washes, and sometimes chlorine. Then it's dissolved in a high salt media with either yeast extract, carbon, or casein-derived amino acids, glycerol, etc. And the option to that is to use this micro-drilling apparatus to bleach the adhesion. So here's a map of worldwide salt basins around the world that you march all the way from the Indian to the Alps Sea to the Cenozoic, our sort of current epoch. And thanks to Alex Gelman, we compiled a list of these. Some of these are still working on compiling a list of these and associated mines. And at first, I was interested in requested samples of Ordovician salt from the Canning Basin in Australia from my friends at University of Western Australia, but they didn't have any in their collection, and even though it's this huge, giant formation, and I had to go talk to the petroleum company there. But anyway, I don't have to do that anymore because their Ordovician formations evaporates in North America and Canada and the US. And likewise for the Silurian and Carboniferous, Triassic, etc. And so we're doing an expedition, great Paleozoic evaporate expedition with my colleagues from lab are joining me, and photographer Ashley Clark, and our county researcher Alex Gelman. And George says he thinks he can get paid for our expedition, so maybe we get like a Winnebago or a hot air balloon or something like that and go to the salt mine and back to Crystals until we get a sufficient collection. And there's benefits to evolutionary biology, hardly provable algorithms of evolutionary biology that predict the evolution of genes across geologic time. We can check those, and it would be useful anyway for evolutionary biology to track for the mutations across those hundreds of millions of years. But also, there are insights in search for extraterrestrial life. These are the seeps, the water seeps on Mars that have been recently discovered of Linnea. And NASA has analyzed the spectra of the salts that like the water comes out and it's brine, but wouldn't be water on Mars, and immediately volatilizing the vacuum Martian atmosphere with salts, and the salts gave us spectroscopic information, spectroscopic data, which is chlorates, poisonous to most organisms, but for several photospheric organisms. And anyway, with that formula, we can build a model for the Oceanus Borealis, the 3.8 billion-year-old ocean on Mars. So I want to build a 10,000 ocean model, so I'm going to titrate from the salt samples from the organisms that I'm growing from the Overdickian and the Silurian and the Cambrian, and over the 10,000 or maybe more, move the formula to the recipe of the Martian ocean, see where the terrestrial extreme organisms, and we can give them, if we do it in Antarctica, we can give them cold resistance, we can give them genes so they can metabolize for chlorates, we can give them genes, hopefully, after April, we should be able to give them genes for radiation resistance. So we can push survivability of the terrestrial organisms more than Martian. And the goal is, of course, this is a garden of biologicals. We want to create a Martian simulant seed. So why is this art? It's because it's a garden, because I want to make flowers for the planets. And I'm not suggesting that we need to deposit these modified organisms on other planets, even though they really would embody one of the most enormous landscape architecture projects that we dreamed of. We would thoroughly investigate the locations for pre-existing life, but it won't hurt to have the toolbox to do the job when it comes to money. So these organisms don't only come from salts and then mines on Mars, but come from some other places in the solar system too. This meteorite fell on Mahans, Texas, in 2009, I think, something like that. And it was 40 feet from these kids who were playing basketball, who eventually got $23,000 for this piece of rock that fell out of the sky. But they found salt in this rock. These guys from Johnson Space Flight Center went right there as soon as they heard about it. And lo and behold, they found liquid water inclusions in the salt crystals in the Mayan meteorite. And they did no biological investigation. They did not do 16S PCR, but they did analyze the iodine isotope content, iodine 149 over time, like the clock, decays into xenon 129, and they found this water in this crystal could be 4.7 billion years old. That's older than the Earth. And there's another meteorite, similarly old, with salts and of Zag inclusions, which landed in Morocco in the same few years. And both of these meteors, and some 70% of the meteors that hit the Earth, are supposedly from this asteroid called Hebe, like Hebe. She's the goddess of youth. She's in this really long orbit. Yeah, doesn't come by very often, but what she leaves, what falls off, hits the Earth. It's in this perfect resonance, like transfer to the Earth. It would be amazing now, because see, even there are other planetesimals and planet-sized objects in the early solar system that landed where they could develop in liquid water that delivered materials back and forth. So wherever Hebe's water came from, there are other salts in the solar system. We think this one being Enceladus, because of the internal. One more thing I wanted to say. Yes. Wouldn't it be amazing to see if we find living materials in these salts from the primordial planetary nebula? Yeah, would change everything. That's amazing, and that's something we should definitely search for. And there are other exophiles to be found, possibly on the ice planets and Enceladus that has a liquid ocean under it, and Ganymede and Europa. So that's it. And I want to thank all these people. Tom Schwartz, my MIT guy, Tom Bork, the reactor utilization officer, Andreas Mershwitz, and Victor Adams. Thank you very much. And James Monahan, especially James Monahan, from the Eastern International Cycle in Kentucky, Alex Jones, for excellent research in honoring Capricorn and Koval for piloting filtration, reactor filtration, and then Mike Napolitano, not only for his contribution to the reactor, but other projects, and Seth Shipman and Jeffrey Bauer, they're like my gurus, they're my benchmates, they're always making sure I don't blow up the world. And John Scheinman, also at Harvard Beach, Paul Regginato, who's here, participating in the reactor project, and Stephen Wolfel at University of Heidelberg, actually Clark, who's photography on the position Harry G. Lewis, and helped pilot the reactor project back in 2011. We started to get way too well with bureaucracy now to solve some of the problems, and because we solved some of those problems, we're able to go faster now. Michael Sismore, who all participated, and then Church Lab in Harvard Genetics, and Thomas Schwartz in MIT Biology, who put up with me for many years, and MIT Nuclear Reactor Lab, and the Beast Institute. And thanks a lot. And BioChanges. Questions?
So from the salts,
Yeah,
is any of it actually bring that to life?
they have. And some of the organisms they brought back to life have never been seen before. That doesn't mean that they don't exist. They just haven't been discovered yet, or they're extinct.
How far back?
Ordovician, that's 500 million years. And I think we can go further.
So I think this question wasn't like, I think why do you want to bring them back? I mean, isn't that always the
you hear what I said about the toolbox too? I mean, we need those tools. They have proteins and genes that we can use that go directly into the toolbox. Unless you're going to be really evangelical about it and say, you know, I mean, what doesn't belong? Because we destroyed it, or because some comet destroyed it, because some accident of atmospheric concentration changed the Earth into an ice planet, and further decimated the population. But all of these things are total accidents. It's like it's natural. Sure, it's natural, but so's Moby Dick and hurricanes, and, you know, tidal waves and tsunamis. And then if you look up art in the dictionary, it says opposed to nature, and for very good reason. Any other questions? I'll be kind. Yes, yes, they do, but they're by and large genotypically and phenotypically distant. The existing hemoplasm they're most closely related to.
So the hemoplasm.
They're both. This is only several experiments have been undertaken. You know, there's a handful of experiments, and mostly isolated coordinations that have unified that. No real survey. Yeah. They occur in all those places that I showed you, but in hot springs and deep baths, you can see on the terrain itself splits and
they're extremophiles in the cranium.
well, except that most organisms on Earth is living in the same environment are actually how they did it.
So they're the name is
Then we are air breathing freshwater creatures. We are the extremophiles. So I said people would do it, but what they realize, but they
But then you can speculate.
we didn't have any genomics is just coming in 1994. We're going to answer those questions now. Our first experiments will be undertaken in April. We think that's our plan. Pardon?
You didn't freeze them at the time?
Yeah, many times in my mind, actually, they're samples we made using things that possibly you know. But we were documenting carefully. We have all these past documentation, and it's actually published in the University Cycle in regular newspaper in Munich. There's a story published about it. Yes. That's true. I think the new news on today, back to international collaboration. So the sequence of genome like we see in Apple, called Malacivisia in Kazakhstan, the genetic evidence shows a UC ancestor. Now it's Domestica, all domestic apples, and the transcriptome, and we compress Wikipedia into first the top 50% of the species pages of Wikipedia into 100 megabytes, but my collaborators in Israel, Joshua Spitzer, has just announced this morning that he's got down to 37. That's pretty cool. Paul, but now also slightly assisted. So we're worried about error correction right now. It's so precisely compressed that it's like even in medical, it's like I have another option for the further compression after I get the DNA form. But then again, it's like if you make one error, then all of the encoding falls apart underneath it. So we need the error correction. You need the error correction bits, bases in the DNA chain that 2000, you want to beat the natural error rate, recurring error rate of standard laboratory techniques and radiation, and to do that, we're thinking one additional base per 1,000. And then we're going to chip synthesize and grow polyclonal catalysis. And each one of the polyclonal catalysis would contain part of the total information. And I have collaborated with the USDA, and now we have a clone. We grafted Malassezii onto disease-resistant dwarfing rootstock, which will give me trees that never get over 8 to 12 feet tall, that I can keep in containment in special area, refrigerated area, because they're temperate plants. And to these trees, macro bonsais, you'll graft our shoots from my polyclonal cups. And I have, if you have another half hour, I can explain that construction strategy.
Can you just clarify? It's like taking the big information and putting it in.
Not all of it could be only 50% of this is for you. This is really ridiculous. You know, I criticize research director of personal intelligence. People who made these messages and made us all have to be like nice people with scientific peace-loving, with scientific attitudes, and all terrible lies and all the censorship and so what is knowledge? See, that's kind of part of this project. It's like, because everything is the reverse of what it should be, you know, and the reason it's should be forbidden is because it includes things like anal sex and human penis size and Justin Bieber, for Christ's sake.
What is the idea?
But I won't censor. You will not be protected by censorship by Joe Davis, I promise you.
Please don't.
Okay.
So, but the idea is to essentially encode the information into the DNA that then replicates and stays alive.
Yeah.
Is that the idea? In
In the genome of the
the
apple.
genome of the apple. But it goes hard in time.
We're lucky that lifespan of apples is under 50 years, and the mutation is that's everything that is 40 years. Well, obviously, for rate reasons. Like, where are you going to put? I mean, it's sort of like I spent 25 years putting all different kinds of information in the genomes of various organisms, including E. coli and the eyes of flies and the ears of mice, and I realized that all information will eventually be contained in this archive because it fixes itself, because you can make hundreds of billions of copies overnight, because we are just unraveling these amazing operations. I mean, this is the kind of development, this increase in operational increase, and the information handling capacity of DNA is increasing at such a rate that it's fair to say that you should think that this kind of technology replacing the internet eventually. Yes, sir. Yes, ma'am. Yes, sir. Yes, sir. You probably have better radiation spectrum, but they're depending on how deep you go, because see the surface of the so like when you go high, you get more radiation. Probably when you go deep, you get this is less of the I would have to say that if you wanted to, I mean, you know, the idea is to get people to send out, contaminate the whole universe, right? To bring the universe to life, right? To solve the Fermi paradox. That's what art has always wanted to do, to bring things to life, to change this kind of living material into this other kind of living material, or this animate object into an inanimate object, and vice versa. That's that dream. Remember, all those legends, all those stories came from the foundation of civilization, when we had first domesticated plants, when we altered, and livestock, when we altered the natural world. We realized we had the power, and so we wrote stories about, you know, Midas and Medusa and Daphne turning into a tree and Moses throwing down his staff and turning it into a snake and all that stuff. And Lot's wife into salt and the whole transformation of the universe. It's the ego dream. And like the most exciting idea about animation, this transformation, this search for extraterrestrial intelligence, because all we're doing is asking a question, and by asking a question, we're trying to bring the whole universe to life. It's cool. Yes, sir.
So when you're sending out in the future flowers for plants, are you going to film the signature? Have you signed it?
Oh, no. 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 I am not one of the top 50 most visited pages on Wikipedia. Who cares? Just at us, dude. You have one life. You have one chance to do something that's really amazing, to change the world, to make dreams come true, right? You just go for it, right? You take up their dreams and you weave them into fine fabric. That's what you do.
I was speaking about you were saying about this desire human things. So how do you feel about robotics then? It's not
You're talking to the person who's like got significantly strange steel. We're going to make sure that there's no geographical variation. If there is, we'll correct it. But we're doing the whole thing. It's interesting. The genome of apples, domestic apples, would probably go to any with many copies. But the wild ancestor apple is a diploid. It has a lot of extra room. Well, by that time, we'll be written into pussy willows and grass on the field and the yellow rose of Texas and bluebells. I have this friend at University of Montreal. He's an evolutionary biologist, and he's a PI. He's got postdocs and he runs a lab. But a couple of years ago, he simultaneously went and got his master's in dance. It's his ambition is to apply what he knew. There are two popular forms of customary forms of notation for choreography. So when we write an algorithm and we're going to apply it to DNA, we take DNA and we interpolate it as a series of numbers. So to the choreography and the choreography is series of numbers to the guy where his algorithms and evolved with anger. Yeah, it's too late to stop our interference with evolution because we're not stupid. If we were stupid, we would, we'd have smallpox and everybody would already be dead from AIDS. That's the nature's way. We're going to defy it. We could just set it up, use my friend Francois' algorithm to set it up so that it always evolves into the correct version, or at least an improved version.
So this is probably not but say mutates relatively 1 million different combinations that would all say that same things. They're all not So the fun communications, but as long as it's sitting down with the baby girl and the safe thing, I'm fine with that. I don't know what the is.
Yes. What's there left to learn? Oh, jeez. Well, one thing we can do is figure out how to make a giant magnetic field, or deflect asteroids with lots of frozen gases and liquids. But however you look at it, terraforming isn't like just bringing a planet to life is a far cry from making a Club Med. Yeah. I have this project with black holes. It's very cool that you don't have time. Thank you very much, ladies and gentlemen.